Hello, I'm Hannah Donnett with the Collaborative on Health and the Environment. Che enjoys bringing you the latest environmental health science through our partnerships, calls, webinars, science serves, publications, and social media. I would like to welcome everyone to today's webinar titled Endocrine Disrupting Chemicals, Effects on Mothers and Interactions with Lifestyle. This webinar is sponsored by the HEEDS Young EDC Scientist Showcase and the Che EDC Strategies Partnership. Our moderator today is Sarah Howard, founder and manager of diabetesenvironment.org. We'll leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentations, our moderator will read out questions for our speakers to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On our webinar page is linked to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speakers. This webinar is scheduled to last for 45 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Sarah.
Thanks, Hannah. Hi, everyone, and welcome. As Hannah mentioned, this webinar is part of a series called the Young EDC Scientist Showcase. It's sponsored by HEEDS, which stands for Healthy Environment and Endocrine Disruptor Strategies, together with Che, the Collaborative on Health and the Environment. So the HEEDS Mentoring Working Group initiated this series as an opportunity for early career scientists to present their research. And this is our fourth webinar in the series this year. If you want to sign up for announcements of future webinars, you can go to heeds.org and sign up for the HEEDS newsletter, and we'll announce them there. So today our topic is endocrine disrupting chemicals, effects on mothers, and interactions with lifestyle. And we'll have two speakers, followed at the end by a question and answer period. Our first speaker is Alyssa Merrill, who's a toxicology PhD student in the Department of Environmental Medicine at the University of Rochester School of Medicine and Dentistry in Rochester, New York. She'll talk about how pregnancy is a critical window for the effects of endocrine disrupting chemicals, or EDCs, on the mother's health. And then our second speaker is Jessica Hartman, PhD, who is a new assistant professor in the Department of Biochemistry and Molecular Biology at the Medical University of South Carolina in Charleston. And she's going to discuss how diet and exercise can influence the toxicity of EDCs. So Alyssa, we are ready, and I will hand it over to you.
Thank you so much, Sarah, for that introduction, and thank you to everyone for joining today. I'm really excited to tell you a bit about my ongoing thesis research investigating whether pregnancy is a critical window for endocrine disrupting chemical effects on maternal endocrine and metabolic health. So as Sarah said, I'm going to be talking about endocrine disrupting chemical, or EDC, exposure today. Now, just as a refresher, an endocrine disrupting chemical is any chemical that interferes with hormone activity. Now, this includes the production, secretion, transportation, metabolism, binding action, and/or excretion of endogenous hormones. It's not surprising that given a definition so broad, an EDC can affect nearly every organ system in the body, from the nervous system to the digestive system. And since the term was coined nearly 30 years ago, over 1,000 xenobiotics have been recognized to have endocrine active properties. These include compounds such as personal care products and pharmaceuticals, to pesticides and metals, as well as a variety of other compounds. Now, historically, our labs, as well as many others, when thinking about the effects of gestational EDC exposure, have focused on fetal health. If maternal health is considered at all, it's classically viewed in terms of reproductive capacity, as seen in the figure on your right, which shows that the measures are usually things such as uterine weight and number of viable fetuses. However, it's becoming increasingly recognized clinically that pregnancy is a critical window for maternal health, both during pregnancy, with there being unique diseases that arise, such as gestational diabetes, as well as for long-term maternal health outcomes. But what makes pregnancy such a unique critical window for maternal health is that pregnancy is coordinated by predictable shifts in hormones. And these shifts in hormones are necessary to ensure a healthy pregnancy for both the mother and the fetus. The graphic that I'm showing here is looking at some hormonal surges that occur during human pregnancy. And you can see that we have surges such as the linear increase in estrogen and progesterone throughout gestation. When these finely tuned hormonal changes are perturbed, as in the context of an EDC exposure during pregnancy, this can lead to the diseases during pregnancy that I showed previously, as well as then subsequently altering long-term maternal health risk. So my research takes a two-pronged approach looking at maternal metabolic health. I investigate both maternal metabolic health during pregnancy as well as long-term. So first, I'd like to tell you a little bit more about gestational diabetes and weight gain effects during pregnancy from these EDC exposures. So the prevalence of gestational diabetes is on the rise. The graphic on your left was published by Zhao and colleagues in 2018, and they looked at nearly 30,000 women from the US National Health Interview Study. And what they found is that over the course of a decade, so in this instance from 2006 to 2016, the prevalence of gestational diabetes rose about 3%. Now, 3% might not seem like a large increase, but when we consider that this means roughly 1 in 10 pregnant women has gestational diabetes, this is a very large amount of individuals that are affected. What's also interesting about this study is that they further broke down the increased prevalence of gestational diabetes by body mass index classification, or BMI. And what they found is that, as seen in the figure on your right, regardless of the BMI classification, there's an increase in prevalence of gestational diabetes. So this led to the question as to what might be other factors contributing to this observed increase. So this is where I took a look at EDCs, as singularly in a variety of epidemiological studies as well as rodent models, it's been observed that EDCs lead to hyperglycemia and increase in gestational diabetes. Now, these include EDCs such as bisphenol A and bisphenol A substitutes, such as bisphenol AF and bisphenol S, as well as pesticides such as atrazine and other organochloride pesticides, and even phthalates and parabens. What these studies did, though, is they only looked at the association with single EDCs. But single EDC exposures are not the reality of pregnancy, as illustrated by this graphic that was published by Woodruff and colleagues in 2011, which looked at a subset of women from the NHANES study. And they measured different EDCs in a variety of chemical classes, such as organochloride pesticides, polyaromatic hydrocarbons, and phthalates. And what you can see from the figure is that regardless of the woman, they're exposed to a multitude of EDCs and a variety of chemical classes. And what's important to remember when looking at this figure is this is only a subset of EDCs being sampled. So think about the sheer magnitude and multitude of EDC classes that women are exposed to during pregnancy. So in order to account for the fact that women are exposed to multiple EDCs during pregnancy, I utilize a mixture of EDCs as shown on the left. And the mixture of EDCs that I utilize all target the hypothalamic pituitary gonadal, or HPG axis. And the EDCs that I work with are bisphenol A, or BPA, an ER ligand, atrazine, or ATR, an herbicide that's been observed to modulate aromatase activity, perfluorooctanoic acid, or PFOA, that decreases estrogen-producing enzymes in the ovaries, as well as TCDD, a persistent organic pollutant that can block ER-alpha activity in the ovaries. And what I hypothesized is that these multiple EDC hits during pregnancy will disrupt the HPG axis, leading to an increase in blood glucose or hyperglycemia, both during pregnancy and postpartum. But the first question that I had when looking into this is, what does this mixed exposure do to serum estradiol levels during pregnancy, as I'm looking at a mixture that targets the HPG axis, which produces estradiol levels? So I exposed to those 4 EDCs that I mentioned on the previous slide at dosages that have been observed to be no adverse effect levels. And we exposed beginning at gestational day 0 in C57 Black 6 mice. And what we then do in order to sample the potential effects on serum estradiol concentrations is we start sampling serum estradiol levels at gestational day 13.5. The figure that I showed you previously looked at hormonal surges during pregnancy in human pregnancies, which has a linear increase in estradiol throughout gestation. However, in rodents, we don't have a linear increase in estradiol. Rather, the surge in estradiol doesn't begin until about midway through gestation, which is why we begin sampling at approximately gestational day 13.5. Then we try to get the peak at gestational day 17.5, and then the subsequent decline that occurs immediately before birth. What I observed is that pregnant mixed dams had lower serum estradiol. As you can see in the figure here, looking at the green line with the squares, mixed dams had lower serum estradiol throughout the course of gestation. This is of concern because low serum estradiol levels in epidemiological studies have been associated with an increased risk of gestational diabetes. So the time point that I didn't mention on the previous slide was gestational day 16 and a half. And we sampled at gestational day 16 and a half, as this is the time point that's been observed in rodent models to correlate with the second trimester of human pregnancy, during which point gestational diabetes testing is commonly done. So in order to further assess potential metabolic perturbations in these pregnant exposed dams, we utilized a glucose tolerance test. And the nice thing about a glucose tolerance test is that it translates nicely into the rodent models, as this is also the test that the American Diabetes Association recognizes as being sufficient for gestational diabetes testing in humans. So how we take this over into the rodent models is that we fast the dams for 6 hours. At the completion of the 6-hour fast, a fasting blood glucose is obtained. Immediately following the obtainment of this fasting blood glucose, we inject with 2 grams per kilogram of glucose intraperitoneally, and then subsequently measure blood glucose levels at 15, 30, 60, and 120 minutes. What we found looking at the top portion of this figure is that mixed exposed pregnant dams had hyperglycemia during the glucose tolerance test, as seen in figure A, as well as an increase in glucose area under the curve, or AUC, in figure B, which is the gold standard metric for analyses of a glucose tolerance test. Now, to determine whether or not pregnancy was the critical window for this acute mix exposure, for 16 days, we also exposed non-pregnant female mice to the mixture. And as evident in figures C and D, we did not see any difference in blood glucose in the non-pregnant exposed female mice. Throughout the course of this 16-day exposure, I also measured weight gain. And what I observed is that in the pregnant mixed exposed dams, we had an increase in total gestational weight gain, where we did not see anything in the non-pregnant mixed exposed female mice. At the completion of the glucose tolerance test, we obtained a wet weight visceral adipose measure and saw that in pregnant mixed exposed dams, on top of having the increase in total weight gain in the middle figure in figure B, we see an increase in visceral adipose weight. Oppositely, in the non-pregnant mixed exposed female mice, we see a decrease in visceral adipose. Now, concurrently with weight gain measures during the course of exposure, I measured food consumption and did not see any difference in either the pregnant mix or non-pregnant exposed groups, indicating to me that this increase in weight gain and visceral adipose observed in the pregnant mix exposed mice might be due to an alteration in energy expenditure. We also, because of this hyperglycemia and increased weight gain that we observed in the acute pregnant mix exposed dams, sent the serum to the Mouse Metabolic Phenotyping Corps at the University of Cincinnati, where they performed lipid panels. And what we found is that in figure A on your left, though we had no change in total cholesterol, what we saw in figure B in the middle is that we had an increase in VLDL, or very low density lipoprotein, which is a negative species of cholesterol associated with atherosclerosis and poor cardiometabolic health outcomes. Interestingly, we saw a decrease in low density lipoprotein, or LDL, which is what we commonly consider the bad form of cholesterol. Now, it has been observed in human studies that lipoprotein lipase, which converts VLDL to LDL, is altered during pregnancy. But at this time, I don't fully know what perturbations might have been occurring to lead to this difference in trend between the VLDL and LDL. I mentioned previously that I'm going to take a two-pronged approach to looking at both maternal metabolic health during pregnancy as well as long-term. So now I'd like to switch gears to looking at long-term maternal metabolic health outcomes. Gestational diabetes is not only a concern during pregnancy, but as evident in this meta-analysis published by Bellamy and colleagues in 2009, women who have gestational diabetes have an over 7-fold increased relative risk of developing type 2 diabetes later in life. So in order to assess whether this could potentially happen from our gestational mix exposure, we once again exposed to our mixture during gestation. This time we also exposed to the singular EDCs that comprise our mixture. Exposure ceased at birth, and then following birth, we allowed dams to continue ad libitum regular rodent chow for 6 months postpartum. At 6 months postpartum, we then ran another glucose tolerance test. And what we found in figure A is that mixed exposed dams had hyperglycemia throughout the glucose tolerance test. When we further looked at the AUC, only our mixed exposed dams in figure B had an increase in AUC from the glucose tolerance test. What's also particularly interesting about the mixed dams in this group is that at the completion of the glucose tolerance test, they have a very elevated serum blood glucose, nearly double that which was observed in any single EDC or the vehicle treatment group. Now, concurrently throughout this 6 months postpartum waiting for testing, we measured weekly weights. And while we didn't see any variance in weekly weight gain, we have quite a large variety of spread here, which might be why we didn't pick up on any differences in weight gain. Once again, since we saw an increase in glucose throughout the glucose tolerance test, we ran postpartum mixed exposed DAMS serum lipids. This time we ran them in-house utilizing ELISAs. And what we observed is that we see an increase in total cholesterol, as well as an increase in LDL, so that negative species of cholesterol. And on top of it, we also observed an increase in free cholesterol as evident in figure C, indicating to us that mixed exposure causes long-term dyslipidemia in our pregnant mixed exposed DAMs. Now, in conclusion, what I observed in my acute study is that mix reduced serum estradiol concentrations during pregnancy. I also saw that glucose processing was only altered in pregnant mixed exposed DAMs, as evident by the hyperglycemia in our glucose tolerance test. Furthermore, mixed exposure led to increase in gestational weight gain and visceral adipose, as well as an increased VLDL and decreased LDL cholesterol species, allowing me to conclude that pregnancy is a critical window for mixed exposure. From my long-term metabolic health study, I saw that mixed exposure 6 months postpartum altered glucose processing, as evident by our hyperglycemia and our glucose tolerance test. I also observed an increase in total cholesterol, LDL, and free cholesterol, showing that dyslipidemia is present as well. However, in this case, I did not see any alterations in weight gain. And for future directions, I'm curious to look at whether or not these altered serum lipid panels are due to non-alcoholic fatty liver disease, if we see any alterations in body composition from mixed exposure, so further exploring that increase in weight gain and visceral adipose that I observed acutely during pregnancy, as well as investigating whether or not mixed exposure led to insulin resistance, which is why we saw the hyperglycemia in our glucose tolerance tests. At this time, I would like to thank both Dr. Corey Selecta and Dr. Sobolewski for all their assistance with my project, as well as the other members of the lab and our collaborators, the James Todd and the Susie Arjo labs.
Great, thank you so much, Alyssa. While we're waiting for our next speaker to pull up her slides, I would like to remind everyone to start putting your questions in the Q&A feature available on the menu bar at the top of your window. We will begin with the Q&A session after our next presentation.
Okay. Well, hello, everybody. My name is Jessica Hartman, and I'm, as mentioned, I'm a new assistant professor at the Medical University of South Carolina. I wanted to start by thanking the organizers and Alyssa, who just gave a really exciting talk. I'm looking forward to discussion. For the invitation to talk today and let you guys and share some of our recent data on exercise and diet and how they affect toxicity from chemicals. So let me see. There we go. So these questions really point to a broader focus in my lab, which is trying to understand in an exposed population of people who are all having the same exposure to the same dose of the same chemicals, very few of them will respond. And it's those responders, the susceptible population, that we need to build regulation around. And so we need to predict who's going to respond and get sick and protect them. And so when thinking about what would cause this inter-individual variability in response to chemical exposures, I've really thought about how that might be impacted by changes in how we take up the chemical, how it distributes in the body, and its mechanism of action once it's there. And then I've really thought about how metabolism itself is probably at the center of a major driver of this inter-individual variability in response. And so I think that there are a lot of factors that can play into how one person's metabolism will differ from another's, including environmental factors, which I think of as anything we can't control. So chemical exposure, sun exposure, et cetera. Lifestyle factors, which includes diet and exercise, which I'll talk about both today, and of course, genetic factors. And so thinking specifically about the lifestyle factor, exercise, that's what I'm going to talk about in this first part of my talk. This has really been a question that has fascinated scientists for some time about what is the difference between a couch potato and a gym rat in terms of metabolism. And John Halsey was a pioneer in this field, first describing adaptations that happen within the muscle from repeated exercise sessions. And so he and others have showed that mitochondrial content, so the size and number of mitochondria goes up in the muscle. And there's these fundamental shifts in the metabolism that happen in the muscle with repeated exercise. But I got more recently, it's being increasingly appreciated that those adaptations are also happening all over the body in tissues that are not considered to be exercised, although that could be argued. But things like the brain and the liver, you see that some of those same adaptations happening metabolically in the mitochondria. And you also see these systemic protections from exercise, improvements in cardiometabolic outcomes, and also protection from these chronic diseases. And so it brings forth this sort of hypothesis and focus that mitochondrial dysfunction is lying at the center of this. So mitochondria in the body are always kind of on their way from this healthy state to a dysfunctional state, whether it just be as a normal function of aging or with genetic predisposition or with environmental exposures that are known to be especially toxic for mitochondria. And then the presence of dysfunctional mitochondria is kind of a key mechanistic step in all of these chronic diseases, such as cancer, neurodegeneration, heart disease. And so I really wanted to understand how exercise, which is protective against those chronic diseases, might be playing in to affect how the environment is driving mitochondrial dysfunction. So where this whole thing kind of started. And all of the data I'm going to show you today, I'm very excited, all of it will be in the model organism C. elegans, which is the main model that my lab uses. And so it has some pretty great advantages. It is a microscopic roundworm, so they grow to about 1 millimeter in length, full grown, and each animal will have about 300 offspring. So we never really have a problem with numbers in this model. They also only live about 17 days, so we can follow them all along their lifespan, and their bodies are transparent, so we can view what's going on inside their tissues in living animals, which is a great advantage, and I'll show you some really cool examples of that today. So everyone always wants to know, how do you exercise a worm? And I think everyone's got it in their mind that we've got these tiny treadmills, and it's maybe not so complicated. So if you don't spend all your time looking at worms, here's what it looks like. The worm lives on an agar petri dish, and when it crawls on that agar, it forms this beautiful sine wave motion. If we put that same worm in liquid, the worm, instead of an S shape, it forms a C-shape motion, and its motion is quite vigorous compared to the plate. And so we have, along with my collaborators in the Driscoll lab at Rutgers, we've done a lot of experiments to quantify how much more difficult it is to swim versus crawl. And it turns out that they put out about twice as much heat during the swimming motion versus crawling, so they really are burning energy. So we have them do this swimming for 90 minutes twice a day over 6 days. And so, as I said, the worms live about 17 days, and they start swimming on day 2, which is also when they start producing offspring. And they swim twice a day for essentially their entire reproductive adult life. And so they, those 12 sessions over 6 days end pretty much at the end of reproduction. And then we follow how are the worms doing as they age after having undergone this long-term swimming exercise. And so the first data I want to share with you is about how their mitochondria were doing in their muscles. So as I mentioned, it's been known in mammalian, human, and rodent muscle that the mitochondria are one of the key places you can see exercise effects. And so on the left, this image is showing a very healthy mitochondrial network within these 2 muscle cells. And the mitochondria are very abundant, and they're networked in these beautiful networks where the mitochondria fall along the muscle fibers. And so as the worms age, their mitochondria undergo this degenerative phenotype. So going from left to right, the mitochondria become more sparse, so you can detect space between them. And they also become enlarged and dysfunctional. And so if we look at the animals 1 day after exercise, so they've undergone the 6 days of exercise, we look at them the very next day. The exercise worms, about 75% of them are having these 2 healthiest scores, whereas about 25% of the population has these less healthy scores on the right. Whereas their non-exercise counterparts are doing a little bit worse. So they've got a little bit fewer animals in the healthiest scores, and more animals in these very severe degenerative phenotypes. So in addition to looking at how the mitochondria look, we can also look at function. And to do that, we take advantage of the fact that mitochondria consume oxygen in order to make ATP. And so by following oxygen consumption from the live animals, we can look at how much oxygen they're consuming at baseline, and also the maximum output that the mitochondria can give. And the difference between the maximum and the basal, which we refer to as spare capacity. And that's kind of a reserve. If the mitochondria need to make a bunch of ATP, how much space do they have to do that? And so at baseline, there was no difference between the exercise and the control groups. It was a little surprising given the differences we saw in the morphology, but maybe not too surprising because they're just going to consume as much oxygen as they need to kind of like function. But that spare capacities, that reserve, was a lot higher in the exercise animals versus the controls 1 day after exercise. interestingly, if we looked a few days later, that benefit is gone. And so they look just like the control group. And that's true in every organism we've looked at. If you stop exercising, you lose the benefits. So we also wanted to see if these improvements in the mitochondria would also translate to resistance to exposure. So we chose a couple chemicals that specifically target mitochondria as part of their toxicity. The first one is arsenic. So looking at the percentage of animals surviving after 24 hours at different doses of arsenite, what we found is that the control group had a dramatic decrease in survival as the concentration of arsenic increased, and the exercised animals did better. So a larger population of them survived that 24-hour exposure. Once again, as we saw with the functional analysis of the mitochondria, that protection was gone by day 12. There's a little bit of an interesting case with rotenone. So this is it's a pesticide and piscicide, and it's a specific mitochondrial inhibitor. And the worms are quite resistant to it on day 8. So we couldn't see really any decrease in survival there. At any dose, we could dissolve. So we were at a limit of solubility here. But by day 12, we did start to see a decrease in survival of these animals, and the exercised animals were a little protected from those rotenone exposures. So finally, we looked at lifespan, where we followed how many animals were surviving each day, and until all the animals in the population had died. And what we saw is that the control group, as I mentioned 17 days was how long they lived. That's the median. So we're about there in the controls, but those exercised animals did live a little bit longer. And so we started thinking about, okay, but we can extend lifespan, but now the aging field is really starting to think about health span and the number of healthy years you have in old age. And so this got me really interested in following up and thinking about Parkinson's disease. So Parkinson's, there's currently no drugs that can modify or slow the progression of Parkinson's disease, but physical exercise has been shown to improve the course of the disease, and the mechanisms are really poorly understood. Importantly, patients who have Parkinson's toward the end of the disease are not able to exercise due to the symptoms of the disease. So we want to understand how this exercise could help in order to develop new therapies in the future. And so again, taking advantage of the fact that these worms are clear and transparent, we can actually visualize their dopaminergic neurons, which are the ones that are lost during Parkinson's disease, and in humans. And so in worms, they have these 4 dopaminergic neurons in their head. So the cell bodies are here at the bottom, and each cell has one long projection dendrite that goes to the tip of the nose, which is at the top of this. And on the left is a very healthy animal with perfect neurons. On the right, these neurons have undergone degeneration, so you can see breaks in that continuous dendrite. And so looking at control animals, this is an animal that has a break in at least one of its neurons. We saw that about 12% of that population had some damage just as a normal function of aging. And if we gave them that mitochondrial toxicant rotenone, that number increased to about 25%. But amazingly, the exercised animals had a lower amount of damage to their neurons at baseline and didn't respond to the rotenone treatment at all. So that was very exciting. And so for this last couple of minutes too, I hope, we've been talking about exercise and how it results in this. If we think about these benefits coming maybe from an effective positive energy balance, then we wanted to think about what about negative metabolic effects, like from a high fat, high sugar diet, something like that. And so I'm going to show you some data from a couple of students who were my lab mates and collaborated with me on this project to look at high sugar diet and worms. And so you'll notice that this picture looks the same. Instead of exercise over those 6 days, we're giving them either a high glucose or a high fructose diet. And we did this to model the increased fat storage and metabolic disruption. And so unsurprisingly, giving them this high sugar diet, whether it be glucose or fructose, increased their amount of fat. And so here I'm showing fluorescence from lipid droplets in the animal that were labeled with a fluorescent protein. And those increased with either feeding. Fitting with the literature, it also increased oxidative stress in the animals, as reflected by an increase in oxidized glutathione. And with a lifespan curve similar to what I showed with the exercise, it's just the opposite effect now. A normal diet animals are in the black line here, and either glucose or fructose feeding decreased their lifespan. And then if we look at the dopamine neurons, again, this is the cell bodies at the bottom and the projections going to the tip of the nose at the top. But this time, the green is not the neuron itself, but the mitochondria inside. We saw that glucose feeding had this effect where it caused hyperfusion of the mitochondria. So instead of being small individual mitochondria, we had these spaghetti-like fused mitochondria in the dendrites. And so we wondered if they would be more sensitive to a mitochondrial toxicant like I showed you for rotenone with the exercise animals. So in this case, instead of rotenone, we're using 6-hydroxydopamine. This works exactly the same way. It's a mitochondrial complex 1 inhibitor. And so this is a little bit of a complicated plot, but the higher the score, the worse the damage. So as we give the control animals more and more of this compound, we get more and more damage in those neurons. But interestingly, whether they had glucose or fructose, my title is wrong here, but they were protected. They were actually protected from that exposure. I just dropped this data in moments ago. It's hot off the press. So we actually saw protection from these high sugar diets. So to wrap up, as in humans, the exercise improved the worm's mitochondria, and it protected them against chemical exposures, including their neurons. But we were surprised to find that diet-induced fat storage was also protective. So the ongoing work is just trying to figure out how all of this works at the mechanistic level. So I won't say too much here except to acknowledge the people that did some of the work and funding from NIHS, and to say in a shameless plug that we are hiring. So if you're interested, please get in touch. Yep. So thank you so much for your attention. I'm looking forward to the discussion.
Thank you, Jessica. That was great. It was really interesting, both of you. So if you have questions, please type them into the Q&A box, and I will ask them out loud. And so I'm going to start. We have one question. We're going to have till about 1/4 up, so maybe 8 minutes or something. So we only have one question so far. And this is for Alyssa, this was asked earlier, can the exposure concentration of endocrine disruptors used in your experiments, are they associated with the real exposure concentration in women?
So as I mentioned previously in my talk, when we selected these dosages, we utilized compounds that are at their no adverse effect level. So they are representative of what women are getting during gestation. But in particular, we picked no adverse effect level dosages, thinking in a policy format as to if we're being told singularly these EDCs don't cause an effect. Well, when in a mixture, they are causing an effect. Clearly, we need to think about where our regulatory dosages are. So yes, they do, in a nutshell, represent the actual concentrations that it's been deemed acceptable to be exposed to.
Thank you. All right, we have no questions, guys. Come on, we have.
I actually have a question for.
Oh, okay.
Oh, can I ask a question of Alyssa?
Go ahead.
So I was curious how much variability she saw in the glucose and the estradiol between the animals, and if she could correlate what she saw with the glucose tolerance with what she saw with the estradiol levels.
That is a really good point. I have not looked at correlating the glucose levels between the estradiol levels. I had very tight serum estradiol levels at gestational day 16.5, but I didn't look in correlating those specifically to glucose. And I unfortunately didn't realize when I did my postpartum studies So looking at estradiol, how you would need to set that up in order to make an association between estrostate and estradiol levels. So that is something that I want to work on postpartum.
All right, Jessica, we have 2 questions for you. So 1, in the worms, do you think the timing of exposure to the high-fat glucose or fructose diets might influence the response? So the timing.
Yeah, that's a great question. I do think so. We chose to start the glucose and fructose feeding when we did. So when they reach adulthood, first of all, to kind of be able to compare with the exercise regimen, but also to avoid any effects of developmental exposure to the high sugars. We wanted to look at them after development, but that in and of itself, looking at developmental exposures or looking at these supplementations in later in life would almost certainly show us something a little bit different and would be super interesting. Yeah, I would like to get into those details more.
Okay, and we have a couple more now. For Jessica, is it possible or likely that the different outcomes would show detrimental effects? I'm not sure what that means. How about, okay, wait, let's go to a different one. Can maybe, Mary, can you explain what you mean a little more? Jerry Heindel asks, can you use the C. elegans for EDC and obesity studies?
I would love to know that. So I'm working on a new collaboration. Well, I'm working on a collaboration with Chris Casotis, and we're trying to look at some known EDC chemicals. But a lab mate from the Meyer lab did look at arsenic to see if arsenic exposures might disrupt their metabolism, and arsenic exposures did cause increased fat. So among other alterations. So I do think that there's a possibility there, and I'm exploring it currently.
All right.
I can mention real briefly that worms don't have PPAR gamma. So that's both a limitation and a opportunity, I think. They do have PPAR alpha. So we can possibly use them to dissect effects through different pathways.
Interesting. Here's another one, Jessica. Did you look at the offspring of the exercised worms, and do you think they would have the same sort of response as their parents?
We haven't looked yet. I think it's a really cool idea. We just, limited time so far, but that's definitely on the list to try to look at effects. Other transgenerational effects have been reported in worms. So intergenerational and transgenerational. So I think it's possible we could see something. Yeah.
All right, and here we have a clarification of the other question. If the high fat diet appears protective for some outcomes, which is surprising, but other measures could be negatively impacted. So
I think that's likely the case. Yeah. There's some support in the literature from rodent studies that obesity, for example, could be oddly enough protective for some chemical exposures, and I suspect it's going to be chemical specific, maybe stage specific. I don't know. It was surprising that they were protected, but yeah, we expect there to be trade-offs for sure.
Well, isn't it in humans? Isn't it that if you're really underweight, you're more at risk of dying in old age, right? I think some body fat is protective in humans, right? And
yeah, that's fair as well. Yeah.
Okay, I'll stick with Jessica for a second. Is there a comparison between how much exercise the worms were doing and how much a human would do equivalent? Like, are they running a marathon or?
Yeah, we've been trying to understand this. I mean, certainly if you think about the length of the session versus the length of their life, it's a long time that they're swimming. But we don't, it's a really hard question to answer to translate exactly how much. But we know that the gene expression changes that we see and the kind of fold change amounts during the 90-minute, after the 90-minute swim session are comparable to like an aerobic activity in rodents or in people. So we think that we're aiming for some kind of like normal aerobic activity and not some very strenuous long marathon thing.
Yeah, I was wondering if there would be a difference. Like if you have too much exercise, could that be, you know, a bad thing in the
Definitely. So we actually titrated the exercise. That was part of my collaborator, Ricardo Laranjero at Rutgers, did a lot of work titrating how much exercise is beneficial. And then you do reach a point where they start getting sick. So yep, we do see that.
Okay, and would you say that engaging in any level of physical activity is beneficial for people who do have Parkinson's or Alzheimer's?
Yeah, I would say yes. I think that is pretty well documented in the literature and even like among physicians. It's one of the first recommendations upon a diagnosis with Parkinson's especially is to try to maintain whatever level of physical activity is possible. And yeah, there's a lot of debate in the field because there's safety concerns as well as the disease progresses, but it seems to help if patients can do some.
And then real quick, I think this one is for Alyssa. That how did you select the toxicants that you used in this study?
Yeah, so what we did to try to account for the fact that there's so many varieties of different classes of EDCs is that we tried to pick 4 of them. So atrazine's an herbicide, so that's where we got that from. Then bisphenol A is just a very common plastic polymer, so we utilized that. And then perfluorooctanoic acid is representative of PFASs. And then finally, we use TCDD to represent persistent organic pollutants.
So were you thinking at all about how about the effects they have? Because it seemed like when you showed that one slide that they all some had totally different effects and maybe even opposite effects, but then they acted in a mixture, interestingly.
So they all singularly have been associated with both gestational as well as type 2 diabetes when selecting them, but the initial rationale was just to try to get a broad grouping of EDCs to account for as many different classes as we could.
Okay. I'm going to ask, we're over time, but I'm going to ask one more question. Of the 930 or so cells in a worm, how many are muscle?
I was glad that Alyssa got a question because I had to Google it. It's 95 cells are muscle cells. So about 1/10 of their somatic cells. They also have the 930 number doesn't include their germline. But of those 930, 300 or so are neurons. And so it's a cool model also to study neurons. Yeah.
I'm going to wrap it up here. So thank you very much. It was really interesting, you guys. I appreciate it.
Yeah, thank you so much, Jessica and Alyssa and Sarah too. What a great conversation, and thanks for presenting your research. So we're approaching the end of today's webinar. Our video recording will be available on CHE's website soon, and tomorrow you'll receive an email containing a link to the video. The next CHE Generation Chemical Partnership Series webinar will take place on March 9th and is titled Endocrine Disrupting Chemicals and Maternal Reproductive Health Impacts. You can find details on our website at healthandenvironment.org. If you are new to CHE and would like to stay updated about upcoming events or more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website at healthandenvironment.org. Additionally, if you appreciate these CHE partnership webinars bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank our speakers, Alyssa and Jessica, for taking time to present today, and to you, Sarah, for your excellent moderation. Thank you so much for joining us. We're wishing everyone much health and wellness. Have a great day.